The two microtubular structures, cilia and flagella. Both cilia and flagella have 9 plus 2 microtubular arrangement. They have a 9 doublet and 2 singlet microtubules in their structure. Both of these are locomotory structures and they have a common structure. But a few differences are present between the cilia and flagella. The first difference is that cilia are many in number and they are present all over the surface of a cell, while flagella are fewer in number and they varies from 1 to 4 per cell. Cilia are very smaller and they have a length of 5 to 10 micrometer, while flagella has a length of 150 micrometer, that is approximate length. And this cilia generates power stroke and recovery stroke in a cell, and it drives or moves a cell perpendicular to the axis of the cell in the direction in which the cilia moves. If a cilia moves in the x direction, then the cell will move in y direction, while the flagella has a wave-like motion and it drives the cell in the same direction in which direction it forms its motion. Means if a flagella beats in the x direction, then the cell will move in the same direction or the x-axis. Few more points that has to be added with cilia. Cilia can be motile or non-motile. If a cilia is motile, it will have 9 plus 2 arrangement of microtubules. These are larger in number in a cell, and the example include the epithelial cells of oviduct and the epithelial cells of respiratory tract, while the surface of paramecium and tetrahymena has motile cilia. But there are few non-motile cilia present in some organisms, and these have 9 plus 0 arrangement and there is a lack of dynein arm. But this non-motile cilia can be of further two types, sensory and primary. The sensory and primary have different processes and functioning, and these sensory are involved in the sensory function in a cell, while the primary include the signaling functions. Coming to the basic structure of a flagella, that's a locomotory structure and present in all prokaryotes and eukaryotes. The flagella has a filament and the basal body or the basal structure. Filament and basal body are together form the flagellar structure or the flagella, and it is made up of bundle of microtubules known as axoneme. This axoneme has a 9 plus 2 arrangement of the microtubules, where the 9 doublets have A and B ring, and this A ring has 13 protofilament, that is the basic subunit of a microtubule, and the B ring has 10 protofilament, which we have studied previously. The 9 plus 2 arrangement has two singlet structure, where 13 protofilaments are arranged forming A and B separate two rings. The flagella has the basal body, that is forming the base of this flagella, is made up of triplet structure of this microtubules, and it has A ring, B ring and C ring. The A ring has 13 protofilament, while the B ring has 10 protofilament and the C ring has similarly the 10 protofilament. This basal body of a flagella is called as the kinetosome, and this is having the basic structure similar to centrioles. The filament of the flagella has an outer covering of this plasma membrane. Now coming to the basic structure of a flagella. Flagella has two parts, filamentous and basal body. The filament and basal body together form the flagella. This flagella structure has a plus end and the negative end. The plus end is towards the filament and the negative end is toward the basal body. This filament part has the 9 plus 2 doublet arrangement and covered with plasma membrane, while the basal body has a triplet of this microtubules, and during the attachment of this basal body with the filament part loses the C ring. But the polarity of this filament and basal body remain the same, and the plus end is to the outer side and it shows the flagellar movement, while the negative end is toward the basal body part and it attaches to the surface of the cell. The doublet of this filament has A and B ring. A is having the 13 protofilament and B is having the 10 protofilament. Both A and B rings are joined together by a special protein, that is alpha helical protein known as the tectin. Now the basic structure of the doublet of the filament. This doublet structure has 9+2 arrangement, where 9 doublets are arranged serially in a circular way and the 2 singlet microtubules are in the central part. This doublet and this singlet are attached together by radial spoke, while the 2 doublets are attached together by tectin protein. This complete ring of the A ring is having a special arm that is called as the exonemal dynein, and it has an outer and inner arms made up of dynein protein. This doublet together are attached by a special protein that is also called as the linker protein. This doublet and singlet structure together form a special cartwheel structure and known as the 9+2 arrangement and forming the filament of the flagella. Coming to the most important microtubular structure, that is centriole. The centriole is present in all animals, fungi and algae. It's a polar structure and it has a barrel shape. This centriole is always present in all animals and it is having a mother centriole and a daughter centriole, and these 2 centrioles are always perpendicular to each other, and this is polar structure having a distal and proximal end. The arrangement of the centriole has 9+0 structure, where the 9 triplets or the microtubular structures are present, but the special example is the Drosophila, where the Drosophila has 9+0 arrangement, but the 9 rings are doublet in their structure. The basic structure of a centriole has A, B and C rings, and together they form a triplet structure. This triplet structure is attached to the central part of this cartwheel structure by radial filaments known as the spokes, and the central part is having a special protein that is called SAS6 protein. That is a special protein. Initially it forms a dimer, and when 9 dimers together join, they form a ring like structure, or they form a central hub of protein, where these all 3 triplets are arranged. Now 3 important points that has to be remembered about the centriole. The first important point is that in all animal cell, one pair of centriole and MTOC, that is the microtubule organizing center, are together as called as the centrosome. This centrosome is different from chromosome. The second point is that this centrosome is surrounded by an amorphous material called as the centrosome matrix or pericentriolar material. The third point is that centrosome duplicates. We all know that the centrosome duplicates in S phase, but during S phase they are connected by a special protein that is called as the tether, and finally at late G2 phase they split completely. So it can be said that centrosome duplicate in S phase, but the final separation occurs at the late G2 phase.